Hello, I am Programmer 53. Today we will be solving the first problem on Rosalind, so that's counting DNA nucleotides. If you're unfamiliar with Rosalind, it's a free web-based platform for learning bioinformatics. The website provides a list of bioinformatics problems that you can see, so each one of these rows is a different problem. The starting problems cover the fundamentals of programming and biology and are meant to be solved to introduce you to bioinformatics and start the development of your programming skills. Okay, I guess that's a good introduction. Let's start by looking at the first problem, counting DNA nucleotides. Okay, so I'm making the assumption you're most likely arriving with a background in biology and not in computer science. Because of this, I'm going to hold your hand through this process as much as possible, at least on the computer side aspect of it. I might clarify some of the biology as we go along, but the main purpose of this video is for understanding the computer science, not the biology. Okay, so now let's go over the problem approach. How are we going to solve the problem? So the first step is we need to make sure we understand the problem. So is there anything we need to clarify in terms of edge cases? Do we understand what the problem is actually asking us and how that's going to lead to our expected inputs and outputs? Once we do that, we can do the second step of our problem approach, where we solve the problem but with no programming. So you should be able to understand the solution even if you have no background in computer science. And then the next thing we'll do is we're going to pseudocode the solution. So pseudocode, if you don't know, is just a high-level description of the solution to the problem, and it uses the structural conventions of programming, but it's mainly intended for human readability. And this step is really important because it's linked to our fourth step where we're actually going to code the solution. So I'll be using Python version 3, but the thing with pseudocode is it's written it's a high-level description, so you should be able, with a little bit of Googling, to convert the solution into most other programming languages. So anything that's so like C, C++, Java, JavaScript, Ruby, all of that stuff should be fine for you to convert the pseudocode into an actual coding solution. I mean, as long as you don't use anything obscure like Trump script or anything weird like that, you should be good to go. After we code the solution in Python version 3, I will put the solution on my GitHub repository. So if you want to take a look at the solution outside the scope of this video, you'll be able to do that. Okay, so let's get started. Okay, so let's start looking at how we're going to solve this problem. So first step of the problem is understanding the requirements. So let's take a look at what we're given. So we're going to skip this first few lines just because it kind of gives us the definition of a string, which I'll go over later, but it's not important right now. So let's go over these two things. So what we're given. Otherwise known as the input, a DNA string of length at most 1000 NT. Well, we can assume NT stands for nucleotides. That's kind of lazy, but they should have put nucleotides. But anyway, it's at maximum we're going to be given a DNA strand of 1000 nucleotides. And then to help me visualize this problem, we're going to add my good friend Pablo to the mix just to give it a little bit more substance. Okay, so my friend Pablo gives us a DNA strand that's up to 1000 nucleotides long. Judging by how he's worded the problem, we know how big the strand could be. We don't really know how small it could be. We could probably assume that the smallest would be one nucleotide long, but we know that Pablo might try to fool us, so maybe he'll give us a DNA strand of 0 nucleotides, because that's kind of a possibility. So we should account for that in our solution. So we're saying that the minimum number of nucleotides that Pablo could give us is 0, and the maximum is 1000. These minimum and maximum values are known as edge cases. So they're the minimum and the maximum, and then all of the cases between are just normal cases. Okay, and then we'll take a look at the return, or the output of what Pablo wants. So after he gives us this DNA strand, we need to count how many instances of each nucleotide are in each strand, and he wants that information printed out in text format in the order of the number of A's or adenines first. Then he wants C's or cytosines. Then he wants the number of G's or guanines, and then he wants the number of thymines, and he wants it printed out like this. So he's going to give us He's going to give us this DNA strand, and then we need to go through it, and we need to print out the number of instances of each nucleotide, and we need to print it out like this. Okay, so I think we understand the problem and the requirements. Let's solve it with no programming. Okay, so we're at the second step of the problem, and so at this point we are going to solve it with no programming. So as we said before, this is Pablo right here. He's going to give us our DNA strand. It's going to be between a length of 0 nucleotides and 1,000 nucleotides, and we have some sort of special tool or microscope that allows us to easily see each nucleotide and its corresponding location. So we can just go through this and count the instance number of each nucleotide, but we need a way to keep track of that. So in order to do that, let's use, I don't know, buckets. And so what we're going to be doing is we're going to have 4 different buckets, and then each one of these buckets is going to be labeled with the certain nucleotides. So we have adenine, cytosine, guanine, and thymine, and in each one of these buckets we'll just place a marble in there when we see an instance of that nucleotide. So we'll start off with adenine. So we see an adenine, we will put a marble in there, and then we'll go on to the next nucleotide. So we have a cytosine, we'll put a marble in there, and then we'll just continue this simple process for every single one of the nucleotides in the DNA strand. And then once we get to the end of this, at the end of the DNA strand, we'll just simply add all of the marbles in each one of these buckets, and we'll give that total to Pablo. And that is a solution to the problem with no programming. The solution of the problem in pseudocode is going to be very similar to this, so it's important to kind of keep this idea in the back of your head when we go through that. So yeah, let's go and pseudocode this up. Okay, so before we go through the pseudocode, I've made a list of programming concepts you might want to understand before we go through that, so it makes more sense. So I've listed them here. So it's definitely encouraged to look up maybe 1 or 2 YouTube videos on each one of these concepts and make sure you have at least a general understanding of each concept so that you're not scratching your head so much through the pseudocode. Okay, let's do the pseudocode. Okay, let's go on to the third step and pseudocode the solution. So first, let's represent the DNA strand in a form that's useful for analysis. We'll use a string to represent the DNA strand. A string is a list of characters in an ordered fashion. This works out perfectly because each nucleotide can very easily be represented as a character. So A for adenine, C for cytosine, G for guanine, and T for thymine. And the order of the string is simply the order of the sequence of nucleotides. After we make the string, we'll store it in the DNA strand variable so we can use this later. Next, we'll need a way to make our buckets to store the marbles. To do this, we'll make a data structure called a list, or it could be called an array depending on the programming language you're using, which is an ordered set of slots where each slot can store a different type of data. For example, we could store a string in one of these slots if we wanted to, but for this example, we want to use the number data type to represent the number of marbles in each slot, and we're going to set the number in each slot to an initial value of 0, to represent the initial number of marbles in each slot. Next, we'll need some conditional logic to add a marble to the appropriate bucket if we see a certain nucleotide, and it's going to look something like this. If the nucleotide we're looking at, which is just a character, is an A, we need to add a value of 1 to the adenine bucket, which is at index 0. Else, if the nucleotide happens to be a C, we want to add a marble to the C bucket, which is at index 1, and as you can see, each bucket has its own specific index. The code in bold, so all this bold code, will work for one nucleotide, but we want to apply this conditional logic to all the nucleotides in the DNA strand. To do this, we'll need to use something called a for loop. This is a control statement that allows us to do a task repeatedly. If we want to make a for loop to repeatedly do a set of logic, it's going to look something like this. We can break up the first line of this for loop into 2 separate pieces. So the first piece is going to be the for I in, and then the second piece is going to be the range 0 DNA strand.length. The for I in is a way to iterate over all the values in a specified list. This is basically saying, give me a list of values. I is then going to be equal to each value, so I is going to be equal to each value in the list once, and is going to execute a set of logic which is listed below for each value of I. So the second half of this for loop, this range function, is making a list that will start at the first argument, which is 0 in this case, and will increment by 1 up to and excluding the second argument, which is the length of the DNA strand in this case. For example, if the length of the DNA strand was 3, we can plainly see this is not the case. But if it was 3, this would create a list with values at 0 at the 0 index, 1 at the 1 index, and 2 at the 2 index. And after this list is created, the I variable is going to be equal to each of these values once, and it's going to execute the logic below. This structure allows us to execute a set of logic for each slot in the DNA strand, which is exactly what we want. Now we just need to find a way to move the conditional logic up here into the magic that is this for loop. Okay, so the only thing we've changed in this conditional logic is set the variable nucleotide, which was a placeholder, to be a nucleotide at a specified index in the DNA strand variable. So now we've applied the conditional logic for adding marbles to each bucket in an iterative fashion, and the hard part of the problem is basically over. Now all we have to do is make the string that will output for Pablo to read. We can make a string that has the correct output by converting the number of marbles in each bucket. So as you can see, bucket 0 is going to have the number of marbles in the adenine bucket, and we're going to convert each one of these buckets, which are data types of numbers, to strings with the str function, and then we're going to join all of these stringified numbers together with spaces, and that's going to give us the correct output that Pablo wants. And finally, we'll print out the output string with the print function, and voila, we're done with the pseudocode. Great, let's move on to the last step and code the solution in Python version 3. Okay, so on to the final step. We're going to program the solution in Python version 3. So at this point, the only thing I've done is I've copied and pasted the pseudocode we just wrote, and I've pasted it into a file that is .py, so a Python file type. We're first going to start by changing the double quotes to single quotes because that's what Python likes, so let's do that right now. Okay, so the next thing we're going to do is change this. So DNA strand.length. So in some programming languages, there is a length property on the strings that gives us the number of characters in that string, but that's not present in the third version of Python. However, there is a function we can use that will give us the length of the string, and that is it's len, and then you just put the string as your input, and it's going to output the number of characters in the string. And then the next thing we're going to do is change these else if statements to elif statements, because that's how Python expresses else if. So let's do that quickly. And finally, when we do our comparisons, so when we're looking at if a specific nucleotide is an A, a C, a G, or a T, we can't use is, we have to actually use a double comparison operator, which just looks like this, and we're just going to do that for every single one of the comparisons. And this should actually work, and in order to run this at this point, all we're going to be doing is running it with the Python 3 command, and then we're just going to put in the name of our file. So we can just run it, and it runs perfectly. It's going to output the number of A's, the number of C's, the number of G's, and then the number of T's. So that's exactly what we wanted. So now let's test this on one of the Rosalind inputs, and let's see if we can get the right output. Okay, so we're ready to try our solution out on the actual problem. So I've already started this particular challenge. There was a download dataset button here that I already clicked on, and it allows me to download the dataset again if I want to. So I can do that really quickly. And so here's the dataset that it gives me. It's in this .txt file. So I can put this in here. So I already have a txt file in there, but I can use the data, put it in there, and so now I should have access to our sequence. So all we need to do in order to test this sequence out on our code is replace this sequence with the sequence that we've been given. So let's just copy this, just cut it, and then we're going to put it in there. We're going to need a backspace one, and then we can try this out with the same command we used earlier. And it's going to output the correct number of A's, C's, G's, and T's. So now all we have to do is copy this. And let's see if it works. Congratulations, you've solved the problem. We figured it out. Yay! Okay, so now that we've reached the end and we've coded our solution, let's take a look at a little bit of extra credit. So what can we do afterwards? So there's three main things that I encourage you to implement if you have a little bit of extra time. The first thing is reading the .txt file input through programming means. So what I did before was I just copied and pasted the sequence, and then I ran the program again. That was kind of janky. You probably shouldn't do that if you don't have to, especially as the file inputs become larger and they don't look as nice, especially if it's not just a sequence. It's definitely better if you take the input that's located in your .txt file through programming means. The second thing that you might want to incorporate into your code is writing the file output onto a .txt file. There was another method that you might have seen when I was submitting the solution to this problem where I could actually submit a file to it instead of just copying and pasting the answer. I encourage you to try that as well because the outputs are also going to become more complicated as the problems become more complicated. And the last thing that I want to kind of encourage you to do is to make the code more efficient. So there are a lot of ways that you can make the code a little bit more efficient. But the main way that I would suggest is to kind of engage in a little bit of a thought experiment. So if Pablo gives us a DNA strand that is 1,000 nucleotides long and it only contains adenines in it, how many if statements do we have to go through in order to read that entire DNA sequence? It's 1,000. We only have to read 1,000. However, what if by some, weirdo circumstance, he ends up giving us a DNA strand that's 1,000 thymines long? Then how many if statements do we have to go through? We have to go through 4,000. So it ends up taking 4 times as long, or we have to do 4 times as many steps to analyze something that should take as roughly the same amount of steps. It doesn't really make a whole lot of sense. It doesn't seem like it's an efficient solution. That's because it's not. There is a better way to do this. There is a data structure that I encourage you to look up. It's called a map or a hash map, depending on the language that you choose to use, and it makes the process for finding your bucket to put the marble in, it makes that one step every single time. So whether you're looking up an adenine or a thymine, it should only take one step. So the amount of steps it would take in order to process 1,000 nucleotides that are all thymines, it should be 1,000 steps. So that's something I would really look into if you want to improve your code.